What mechanism allows viral capsid self-assembly? Imagine tiny protein pieces floating in a cell, suddenly coming together to form a perfect protective shell for a virus. It's like a microscopic puzzle solving itself in seconds. That's what viral capsid self-assembly is all about. It's a natural process where multiple copies of viral coat proteins organize themselves into a precise, often symmetrical, shell around the viral genetic material. This process doesn't need an external energy source like electricity or heat. Instead, it relies on the inherent properties of the proteins and the physical forces at play. The key to this self-assembly lies in the shape and chemical makeup of the capsid proteins. These proteins have specific shapes that fit together like pieces of a jigsaw puzzle. They also have chemical properties that allow them to stick to each other through weak, reversible interactions. These include electrostatic attraction, which is like magnets pulling together, hydrophobic effects where water-fearing parts hide away from water, and hydrogen bonds that act like tiny glue spots. These interactions provide the energy needed for the proteins to bind together, but they are weak enough to allow the structure to be flexible and correct itself if needed. The process usually starts with a small cluster of proteins forming a stable nucleus on the viral RNA or DNA. Think of this as the seed of the whole structure. This seed then attracts more coat proteins, which add on one by one in a step-by-step process. The viral genome often helps by providing a negatively charged scaffold that draws in the positively charged regions of the coat proteins, making assembly more efficient and specific. As more proteins join, the shell begins to curve and close, forming a complete protective container. But this process isn't always smooth. Mechanical stresses can cause the shell to form incomplete or odd shapes, like open shells or cylinders. Sometimes, the assembly gets stuck in a state that isn't quite right, called a kinetic trap. Viruses often need a maturation step after the initial assembly to become fully stable and infectious. From a weird science view, this is like proteins performing a magic trick, floating around randomly and then suddenly snapping into a perfect shape without any blueprint. Scientists have found that changing conditions such as pH levels, salt concentrations, or adding synthetic molecules can cause the capsid to misassemble or form strange shapes. This shows just how finely tuned and delicate this process really is. Because of this natural ability, scientists are inspired to mimic viral assembly to create tiny nanocages for drug delivery or nanotechnology. They look at how proteins self-organize and try to replicate it artificially. So in the end, viral capsid self-assembly is driven by the specific shapes and chemical properties of proteins, the initial formation of a small nucleus, and a balance of forces that guide the proteins to come together perfectly on their own. It's a tiny miracle happening at the microscopic level, showing how nature can create complex structures with simple rules.